Okay, welcome everybody to this month's installment of the Chalmers AI for Science seminar series. Today we have Matteo Degiacomi from the University of Durham University in the UK. Matteo is originally from Switzerland. He did his PhD at EPFL in Lausanne. He then moved to Oxford for a postdoc with a grant funded by the Swiss National Science Foundation, and sits 6 or 7 years at Durham, first at chemistry, and now he's the tenured faculty at the physics department, and he will give us a presentation on how he uses machine learning to sample protein conformation spaces. So please take it away. Let me...
Thank you so much. And,
just 1 second. You should share the screen. I just need to...
Oh, yeah. Am I sharing?
You should do it on your computer now.
Oh, man.
Sorry.
We start with a bang right away. Second. Oop. Wait, what? See, I'm a computational scientist, as you can see. Now I'm struggling sharing a screen. Zoom. Ah, there you go. Oop. Oop. It's probably this one. Slideshow. Yes, it works. Yay. Right, as I was saying, good afternoon, everyone. So thank you for the invitation. I've been having a great day today in Gothenburg. Right, so this is a story that, as you can see, is about using machine learning to learn something about the conformational spaces of proteins. It actually starts using, it uses a pretext of a pretty old work back from 2013. And when I'll be talking about that, you'll be wondering, yeah, but where's the machine learning? So bear with me. There is a reason why I will start telling you, not directly about machine learning, but about iron. So I really like this story because, so this is my favorite and most hated protein at the same time because I did a PhD on it. So you both love and hate what you do. So iron is a pore-forming toxin, first of all. The only thing you need to know for this presentation is that this is one of the most famous in ancient form of attack between cells. So the mechanism of pore formation essentially requires, in this case, a bacterium to secrete the protein as a monomer. Then what can those monomers do? Well, by themselves, not much. But what they can do, they can assemble to form a complex on the surface of the membrane they want to attack. So you can see an example representation here. Now, this is not actually the final form of the toxin. Once a circular symmetry is assembled, that is here shown, this kind of volcano-shaped protein, there's a conformational change, fold switch, and the protein inserts a beta barrel membrane of the protein, and this is what kills the cell. Depending on the size of the cell, it's either osmotic shock or you have the uncontrolled inflow of ions, and this is what essentially cell necrosis, disintegration, essentially. Now, why am I telling you this? So this is a problem we were faced at the time in 2013. The only thing we knew about this protein is electron density, a pretty low resolution of what's called the pre-pore state, so the volcano, and the pore state, so the kind of disk. And our collaborators in EPFL, so the group from Gisou van der Gut, asked us if we could produce a model, an atomistic model of how the protein looks like in the two states, and perhaps with the goal of understanding how it switches between those two states. And as soon as you see that, I hope you have the same reaction I had. Well, we have the structure of the monomer, but there's no way we can just try to stick 7 proteins together in a circle so that we can make them look either like a volcano or as a disk, depending on how we assemble. There should be some rearrangement. We should figure out how to accommodate while we model. Now, what we're describing here otherwise is a classical example of protein docking, which essentially is, well, you have your building blocks, which in this case is the protein you've seen before. And what we're trying to do is to figure out how to assemble those building blocks into a complex that hopefully makes sense. Now, the issue is that your building blocks, there's plenty of different ways you can assemble them. One is the correct biologically meaningful assembly, and all the others are not. Now, our job essentially is to figure out how to assemble those complexes and discriminating the bad assemblies from the good ones. Now, some of them make obviously no sense, like the pile of Legos you can see there, but there are plenty of assemblies that per se seem okay. and that we should be able to discriminate. Now, if we now think about computationally what that means, well, we need to be able to roto-translate proteins in space. We need to be able to assess their energy. And if we had a very accurate way of manipulating the protein and assessing really accurately the energy, in principle, we should be able to identify between all of the different assemblies you can still see there, which one is the best one. But the issue is that actually we do not have perfect ways of assessing the energies. The landscape of all the possible orientation is a very rough energy landscape. And so that ends up, so being able to rule out those models is hard. But what we can do, we can use construction rules. So if we have experimental data and we can integrate the data in the search process itself, we can actually rule out the, this can help us ruling out some of the candidates that in principle would not otherwise be ruled out. Now, this is essentially an optimization problem. So what is the best way? We have to assemble all our building blocks so that the energy is minimized and experimental data is recapitulated. So essentially, it's a constrained optimization problem on a very rough landscape. Now, the point I'm making here is that the classical way people have been used for many years, including us back in 2013, was to use optimization algorithms. So in this case, we developed a particle swarm optimization engine, which we call POW, which is what we used to solve this problem. So in the previous slide, what you've seen that they were actually inside the electron density, they were atomic models. And those were actually models we produced using POW in combination with the atomic structure of the protein and experimental data. Now, if you're wondering why I'm telling you all this, the point comes in the slide that is about to appear now. So we said that essentially we need to be able to accommodate somehow the flexibility of the protein. So how do we do that? So at that time, what we did is the following. We ran a molecular dynamics simulation of the monomeric protein. And you can see that's classical molecular dynamics of a protein. Applying two laws of motion, let them evolve through time. You get essentially a movie of the protein as it samples its energy landscape. Then what can you do? Well, you can do a classical thing people have done for many years. You can run a principal components analysis of this. This looks very wobbly and noisy, but we can decompose this motion into a linear combination of eigenvectors, essentially, where every direction represents one linear motion. And the good thing about proteins is that there tends to be there's many degrees of freedom, but there's a high correlation in your data. So you can aggressively reduce your landscape without actually losing much information at all. So what you're seeing in the plot there on the bottom right is actually the principal components analysis of the simulation you've seen before. We can preserve most of the variance just by projecting into the image. And each point you can see is a conformation in the simulation. And at this point, why is this interesting? Why is this useful? Well, because we can use this information for docking. What you can do, you can essentially say, well, normally if you're trying to dock proteins, you would just take your subunit and rotate them in space. But now we can augment the search space by combining the search for suitable roto-translations that build the model with coordinates in that space. Thus, the eigenspace is the space is the projection into of our simulation into dimensions that each maximize the variance. So what does it mean? If we want now to build a model, we can pick a coordinate in that space. We can then apply a roto-translation of choice and build a model. And this is, you can send to POW, and POW will identify not only the best orientation of the molecules, but also the best conformation as described by this space here. And this is where we start talking about machine learning. So what is that space telling us? This graph here. So, well, we can see there are some states, but now what we can see is that essentially every snapshot we have from the simulation lands somewhere in the space. What you can do with the principal components analysis, you can actually, just by taking linear combination of, let's say, those two eigenvectors, rebuild something that kind of hopefully looks like the original protein at the how it should look in a specific coordinates in this place. So I could, let's say, point at that position there where there's a blue cross and say, well, how would that look like more or less? In principle, you can take a linear combination of those two eigenvectors at that specific coordinate, and you should get something that kind of looks like a protein. But the protein will look terrible. So principal components analysis is good if you're trying to look global trends in your data, but here fine structure matters. and essentially all the bonds, all the angles, all the dihedrals will be all over the shop. You'll have clashes. It essentially, don't do that. That's the bottom line. So first, this point was, and that's something I've been wondering since 2013, is there a way we can do better than that? And this is actually where the idea of using other techniques comes into play. Principal component analysis is an example of machine learning, is just not a very useful one for the task we have in mind. Yes?
There's a question from the chat now.
Sure.
What is the PCA applied to? Is it I guess the coordinates, the C alpha coordinates? Or?
Yes, so in this case, that was a PCA on the C alpha coordinates, yes. So now it turns out that the PCA is this is what we call a dimensionality reduction. So we are trying to essentially represent some high dimensional space represented by the coordinates, in this case of the C alphas, into some low dimensional space that we can then use for our own purpose. In this case, identify adjacency between different conformations. So what we could not do at the time is we could not pick an arbitrary coordinate in this space and use it to generate a model, because as we said, those coordinates would generate proteins that conformations that did not really look like proteins. Ideally, so in the end, what we did, we just, in our optimization, we pick a coordinate, and then we actually selected for modeling the closest conformation sampled in the molecular dynamics simulation, because that looks like a protein. So we thought, well, is there a way we can actually have a space that we can actually sample as a continuum, as opposed to just being limited to the samples we have explicitly seen? And well, so one of the famous neural networks that actually is capable of doing dimensionality reduction, that's something that's known since the late 80s, early 90s, is the autoencoder. So in 2019, well, we thought, well, can we just use an autoencoder instead? And I mean, I probably don't need to explain that to this audience, but I will just spend a couple of words anyways. So actually, the autoencoder, what it does, it takes some data, tries to compress it through a neural network into some low dimensional space, which we call the latent space. That latent space then gets decompressed on the other side, and what we're trying to achieve essentially is to identify which set of parameters in our neural networks encoding and decoding the data that leads to essentially compression and decompression with minimal loss. If we are able to essentially get on the other side of the decoder something that looks like the input, we have a fantastic way of compressing the data down to a low space and low dimensional space for our purposes. So can we take a protein, compress it, and get on the other side something that looks like a protein? You can train a neural network by, at this point, running a molecular dynamics simulation, sending all your conformations, and what you get is this latent space, which for all intents and purposes we can interpret as the PCA, the eigenspace we've seen before, with perks. So the latent spaces are actually, unlike the eigenspaces, they are nonlinear. So the bad thing is that being nonlinear is very difficult to actually rationalize what each dimension means. So if you're actually looking for explaining what the neural network is capturing, those latent spaces are not, I would say, very useful. But if you want to use them as a means of effectively describing the space in a way that then can be used to generate new conformations, they are useful. If you actually are getting to a situation where now we have the examples of 3 example proteins projecting here, you can also point at any other position in the space and generate proteins that did not exist in a training set, but hopefully are still consistent with it. So this was the naive way of going, and we thought, what is the simplest possible way we can go by? So we did not do anything special there. We literally just said, this is a fully feed-forward neural network. Every neuron receives a degree of freedom. So if we have n atoms, there will be 3 n neurons in the first layer. Literally send it and let's see what happens. Just let's see. And to our surprise, it did work actually. So that's an example of a protein called P24. You can see 2 domains hinging on a flexible linker. Classical thing you would do: take the simulation, split it in a training and a validation set. You only show the training, and then you want to see whether the validation set can be appropriately reconstructed as it gets sent through the neural network. And essentially, what you're seeing on this histogram here is the root mean square deviation of the validation set, comparing it in terms of input and output. So backbone RMSD, on average, if I remember correctly, we're at 1.47, which is actually surprisingly not bad. You can see at the very top some snapshots of our ground truth in this case would be, well, the conformation from a simulation in blue, and in red, what the neural network believes that conformation should look like. So per se, that's encouraging, but there were problems, many of them. And so essentially, first of all, well, it's not because the root mean square deviation is low that the structure is right. So if you think about the energetics of a protein, you can just take two atoms, you can just move them slightly close together. Suddenly, the energy goes through the roof because you end up in the power exclusion principle zone. But in terms of root mean square deviation, it would be exactly the same as moving them slightly apart. So low root mean square deviation is good, but it doesn't mean that that is a structure that actually looks like a true protein. So that was the first problem. The energy was just not there. Then training this neural network was an absolute nightmare, unsurprisingly. And actually, the other issue that was just a slight annoyance, but as we'll see, that led to an interesting development, is that, well, I said the first layer needs to have a number of neurons commensurate to the number of degrees of freedom in the system. So every time I train a new neural network, I need to change the architecture. Every time I train a new protein, I need to change the architecture of the neural network. So the first layer, having a number of neurons commensurate to the number of degrees of freedom of the protein.
When you apply this algorithm, how many latent space do you count at the end? Or conditionals at the end?
Right. So in this case, so for this specific example, that was the first try. We literally just went. We tried several dimensions, but what I've shown there is 2 dimensions only, which is quite aggressive.
And if you increase the number of dimensions, do you expect the sum of these things to be less severe?
So the answer is yes, but only to a certain extent. So essentially, what happens is we'll see a little bit about that more later. But of course, if you try to compress the data too much, eventually you end up in a situation where you don't have just enough dimensions to be able to capture the correlation in your system. But the problem is that if you have a latent space that is too large, what that means is that the data becomes very sparse in that space. And at this point, it becomes more difficult for the neural network to generalize what basically happens in the space between different conformations. So there's a kind of trade-off. You want to compress the data enough, but not too much. 2 dimensions is, unless you have a simulation like the one I've shown you before, where you essentially have a protein with 2 domains moving like rigid body. In a general sense, 2 dimensions in our hands is too aggressive. We have found in our testing, in the next neural network I'm showing you, that 4 or 5 dimensions are ideal. The disadvantage is that you cannot make pretty plots that I can show you and point at the latent space. So I'll be showing you 2 dimensions, but in reality, we use more normally. Yes.
There's another question from the chat asking whether the encoder and decoders were roto-translational invariant.
So when I said that we did the most naive possible thing, they were not. So we literally did the most brutal thing ever, which is take the simulation, align it, which at least cancels out the translation. And well, it's because we can align it that essentially we make it resistant to the problem of roto-translations. But that's as brutal as it went. We did not think about accommodating for that at all, because we know how we can align the data to cancel the roto-translations out, essentially. Now, this was just what we started, but things got better. Don't worry, now we actually get to something more exciting, I think. So how do we solve those problems here? This is actually a work of my collaborator, Chris Wilcox, is in computer science in Durham, then Kat, postdoc in my group, and Sam is currently writing his thesis. And if he pushes very hard, he can submit in December. I hope he's listening. Sam, I'm talking to you. So anyways, what you're seeing down here now is just a schematic representation of a typical autoencoder, exactly what you've seen before. So take a protein, we call it coordinate with the variable x1, compress them, so encode them into latent space, and then decode them and compute the means between them. Now, we first of all changed the way we train the neural network. And we thought we should come up with a way of encouraging the neural network to generalize. And so we came up with a different way of training that you see schematically represented here. And that made a huge difference for us. So let me explain what you're seeing here. So now, what we do is the following. We first of all take not just one example at a time and send it to the neural network, but we train 2 examples. Now, those 2 examples will land somewhere in the latent space. Now, we actually, like normal, take those examples, decode them, and check the mean square error as before. This is still done. However, we also take a random intermediate between those 2 points. The intermediate is picked on as a random point on the line that connects the latent space equivalent of these 2 examples. So take any random midpoint. We decode that as well. Now, that example, so that intermediate does not have a counterpart in the training set. However, what we know, what we would like is that any intermediate should have an energy as low as possible. So what we do, we decode that as well, and then we calculate the energy of that complex using an MD force field. And at this point, the loss function combines both those terms. And this effectively is telling the neural network that not only you should be able to compress the data, but you should also be able to generate intermediates that are anywhere, that any intermediate should be as low energy as possible. So this helps actually making the landscape less rough. And the second thing we figured is that actually the encoder and the decoder, we don't need to actually use classical neurons, but we can actually write instead a convolutional neural network. And now you might have an idea of how you could, how it's done nowadays, typically, a convolutional neural network with points in space. And probably we have not done what you're thinking. So let me, or maybe we have. Let's find out. So what we did is the following. First of all, classical thing, the convolution is an operation that is typically used when you deal with images, at least most commonly. So essentially you have an image and you have some kernels that essentially you will slide through your image. And essentially what those kernels act, they act as filters. When what you're seeing here is the classical Sobel filter is essentially an edge detector. If you pass now those 2 filters on the image you can see here, on the other side, you will find essentially the edges of the image. So now what we can do, we can actually, that's what's a convolution, but the issue is that how do we actually run convolutions on a protein? A protein is not an image. We represent the data so that the convolution becomes an operation we can carry out. So protein is an arrangement of points in space. And at this point, perhaps one thing that you can do, and that one thing that is done, one possibility would be, for instance, building a grid, casting very fine grid, casting every atom in this grid. And at this point, you have the equivalent of an image. Or perhaps what you can do, you can calculate a distance matrix. And at this point, the distance matrix becomes an image you can actually convolve onto. And that's not what we've done. So what we did instead is we thought that, well, actually, a protein is, well, at least the backbone, you can essentially sort your atoms exactly. They are arranged in a chain. If you think about how they are stored in a protein data bank, essentially every atom appears in a certain line with its coordinates. And what you know is that atoms that are consecutive in the PDB are adjacent in space as well. So when you're dealing with an image that has 3 channels, the R, G, and B, you're running convolutions on the red, on the green, and on the blue in 2 dimensions, because the image is 2-dimensional. So here what we have instead is the equivalent of our RGB are our XYZ. And the convolution, instead of being 2-dimensional, is 1-dimensional. So our convolution is essentially doing that. And that is essentially the architecture we put together. And this made the training so much faster and so much easier. And just to show you that it works, I have an example for you, which is the example of this protein here called MERD. So MERD, what you're seeing here is an overlay of 2 crystal structures. It's a protein that has been crystallized. And that's the structure is in blue. And the structure in red is without the ligand. And essentially, coarsely, you can say that you have one domain that moves with respect to the other. So just for description purposes, what we can say is that, well, we can coarsely describe the different conformations the protein can take. This is just for us to describe it as spherical coordinates of the little spherical domain at the top with respect to the long domain to the bottom. So we have essentially elevation and azimuth and a radius and distance. So what we did is we have those 2 atomic structures. We run very quick simulations of the 2 states of the protein, the closed and the open state. And then we submitted our neural network, which we call molearn, in the simulation of the 2 states. And now what you're seeing in the plot on the right-hand side now is, first of all, that's a projection in 2 dimensions. as we said before, we can see that. So that's why we like two dimensions, just for demonstration purposes. Now, every point is where a snapshot of either the open or the closed simulation lands. The color, that's what's called the dope score. So the dope score is a heuristic that is used by software like molearn to basically, it's a heuristic for the energy of the protein. Can be quickly calculated, is good, and is used in homology modeling. So what we did to generate that, the color landscape, essentially, we generated every three-dimensional equivalent of each coordinate in this grid here. If it's yellow, that means that the dope score is terrible. The darker it gets, the more we have something that is good. And ideally, we want a number that's negative. Now, the point, the red and the blue point, is where the crystal structures project. Now, if you look at that, you'll probably say, yeah, but Matteo, it looks like the open and the closed state are in contact. So you probably had your simulation were probably swapping between the two states, right? So essentially, if we now ask the question, where is the intermediate between the two crystal structure, the red and the blue? Well, I mean, one thing you can do, you can draw a straight line in the latent space. Somewhere in the latent space, there will be a structure that we can call the intermediate, but you kind of have an intermediate already, right? Because you've got all those black points around. Now, this is where the things get interesting, because we said that the latent space is non-linear. So in this case, what happens is that the neural network has essentially pushed together conformations that actually in the simulations were not that close together. It basically warped the space to move them together. And to convince you that is true, if we now plot the elevation and the azimuth of the simulation of the open and the closed state, and we plot them one versus the other, what we see is that actually the simulations never interconvert. The closed stays closed, the open stays closed, it stays open. And so if we ask the question, where's the intermediate between them, we actually have no example at all of that. So when we actually draw that straight line and we generate all the intermediates, what we get is essentially this movie here. So this is actually what the neural network generated. The straight line in the latent space it can be something a bit more complicated. In this case, you can see a straight line is associated with this kind of circular motion. Now on the graph on the right-hand side, what you can see is the squares represent the trajectory followed by the interpolation you can see here. And essentially, you can see that you need to go in two directions. That's at least what the prediction is. If you did this prediction with the principal components analysis, you would see the trace represented by the circles. Now, is this right? So we did two things to check. One, we actually took the closed state that stays closed only if a small molecule is bound, and we removed the small molecule. And at this point, the protein spontaneously opens, and it opens like that, essentially, where the neural network predicted it. And the other thing that I wish to say that it was by design, but actually we found out only when we started working on this. There have been crystal structures solved for this protein trapped in an intermediate state, which we didn't know about, and we had therefore also not shown to the neural network, which is great, because now we can check where they are in this latent space, and they happen to be down there. So essentially, what we have here, we think is a way of finding a possible reaction coordinate between the two states. And it's very quick. So instead of having to run microsecond long simulations, so we just had to run sub-microsecond simulation. It was half a microsecond for open and closed states, which with a modern computer infrastructure, you're looking at a day and a half, more or less. And the neural network itself trains in between 20 minutes and an hour. So we can get a guesstimate for a possible reaction coordinate that then can be refined with more computationally expensive methods quite quickly. Yes. Yes. So I'll describe the graph again. So first of all, the light green colors on the graph, you can see that there's two clouds. The one to the bottom left is the closed state. The one to the upper right that is more diffuse is the open state. And they never switch between each other. That is actually what we have shown the neural network. Then the neural network was asked, well, predict me an interpolation between the two. That essentially was done by drawing a straight line in the latent space. And that straight line in the latent space was associated with a motion that casted into this elevation azimuth space is associated with the trace shown by the squares. If you do the same operation, but with the PCA instead, you will get the trace shown by the gray circles. And finally, the dark blue maybe points. I don't know what to call it, dark blue maybe. That is what you obtain if you take the closed states. That is a simulation that to stay closed requires a small molecule to be bound. You manually remove the molecule. And at this point, basically the protein is not held together by that, and it just cracks open. And what you're seeing there is essentially the trajectory of opening from the closed to the open state. You can notice that actually there's way less points, dark blue points in the closed state than there are in the open state. It opens quite quickly. And finally, well, the stars are the internet. Right. So another interesting thing now, just to mention that physics does something good. So adding this extra physics bias helps. If we look now at what we can look at is essentially whether locally the structure of the protein is correct. So in this case, we are coloring the proteins. On the left-hand side, you have the closed state. No, sorry, the open state. On the right-hand side, you have the closed state. What's in between is a prediction. So there's no equivalent in the training set. The color means, well, if it's light color, that means that locally the structure is good. So we're essentially just checking how far bonds, angles, and the hedral are from equilibrium. And we see essentially that the intermediate, if we add physics, has a substantially better structure. So the domain, the small domain that moves as bonds and angles much closer to their equilibrium distance. So we are basically getting better local structure through that. Now, one thing we're trying right now is actually, we're trying several things. One thing, there's a collaboration with Antonia May in Edinburgh. So one thing we are wondering is whether we can use this estimated path between states as a reaction coordinate that can be investigated with other techniques. So one thing we're doing, and that's from Ryan Zhu, who's a PhD people supervised. We have their own simulation of chinolin. It's a 12 amino acids long peptide that essentially, well, we have 16 microseconds. It's a peptide that essentially switches between two states. Either it's this hairpin on the right-hand side, which is folded, or it can be unfolded in many, many different ways. So we're trying to see whether it's possible to train a neural network and project essentially this whole conformation in 2 dimensions and see whether the folded and unfolded states land in different regions. And if they do, whether we can use a pass as a way to describe the transition between the two. So this is actually, it's fresh. So we are still working on that, but essentially this is what we're getting. This is what we're getting at the moment. And the white points is a trail that follows the lowest dope score path between a closed and an open state. And essentially, we see in what we know here is that there's a bottleneck. So you can take any open or closed state. You will always have to go through to essentially the same area. And now essentially the idea is, can we do commit analysis? So transition path sampling is essentially what's happening right now. So hopefully the next time I see you, I can tell you something about whether this technique is actually useful. And that's one thing. And the other thing we're wondering is, so we trained using simulations. Can we train only with experimental data? So we went in the protein data bank, and one of the proteins that is most heavily studied is actually structurally is calmodulin. There are 594 atomic structures. Some of them are NMR, which means that you end up having 1,243 conformers, which happens to be enough to train a neural network. So calmodulin looks like, well, that. If you overlay all of the different conformations, you get that. So it basically domains that can move all over the shop. And what you can see on the right-hand side is a latent space only trained with experiments. And again, very early result, but it seems we can discriminate different states that are known of calmodulin. So hopefully this could be an interesting way of actually either just classifying the states of a protein can take, but also trying to identify possible pathways switching from one state to another. X-ray crystallography and NMR. Yeah, just purely structural data. Just go in the PDB, look for calmodulin, download everything. One thing to mention is that actually those calmodulin structures needed to have some post-processing. Some of them were mutants, and some of them were missing some parts. So we basically model everything back in, and we transform them into a consensus sequence, so that we could actually just train on backbone only. So that's what we did in this case. Again, this is super early, but I just wanted to show it to you, just because I'm actually quite happy to see that some regions have a negative dot score. It's quite promising. How do you know? Yes, that's correct. Yes, that's that.
Do you actually see that any of the NMR structures that actually sample something more than just like variation?
So early analysis seem to indicate that the NMR tends to cluster in a specific region. I don't have one NMR that spans the whole latent space. Yeah. Now, the other thing we wondered is, okay, so up to now we trained the neural network on just one protein or at a time. Can we do transfer learning? So before I've shown you the neural network trained on this protein that works like Pac-Man with a drug. It's closed when it has the drug bound, otherwise it's open. Now, let's go back to the very first simulation I've shown in this presentation. This is the HIV-1 capsomer. So, if we now, well, which recall moves like that. Now, the graph you're seeing here shows us essentially how the performance of the neural network in terms of loss function improves the more we train the network. Epoch, every time essentially you show all the data. What you can see here, unsurprisingly, is the neural network gets better the more it sees data. The color band is because that's on 10 repeats. Now, what happens if instead of initializing the neural network from scratch, we initialize it from a neural network trained on the other protein, D, which looks completely different. So we get that. So essentially, pre-trained network looks faster, which and this actually is replicable even if we train with only 100 examples. So what this is telling us is that essentially there is something fundamental the neural network is learning about how atoms arrange in space that does not need to be relearned every time you train a new protein. And so we could replicate with other proteins. So I'm showing you an example, but we've done that for other proteins, for 4 different proteins. And we have a hunch about what that thing that is being learned is. So essentially, a convolutional neural network in the first layer, it looks for local structure. The more we travel through the layers, the more we go in the deeper part of the network, the more the receptive fields of each individual kernel becomes wide. So then the internal layer are gaining more an awareness of the general structure of the network, of the protein. And now the first layer is looking for local correlation, that in this case is bonds, angles, and dihedrals. And every protein will still need to have bonds at a ballpark 1.5 Armstrongs. The angles should, and the dihedrals, well, dihedrals, we know that you need to still respect the Ramachandran distribution. So those are all things that you don't need to relearn. So at least the first layers can be parameterized quite accurately starting from any protein, and we can see benefit here. So this is what I'm hoping is that eventually we will get in a situation where we can train a neural network with multiple proteins at the same time. At the moment, we are just doing transfer learning just to speed up the way we can converge to a yes.
The local structure features, or there's anything, because the local structure in principle, like as you said, what to expect, so you can just impose it in the generating process instead of having to learn it. So is there anything else, or is it just local?
So there might be something else, but I don't. To be honest, the answer is I don't know. So the fact of local structure that's there. Then if there's any other factors, I don't know. What I'm guessing is that if you and that's something that we're trying right now, actually. So again, hopefully next time I see you. If you do transfer learning on a protein that has a fold that is more similar to the protein you're transferring from, I would imagine that even maybe the second or third inner layer might already be closer to a good set of parameters. So what I would expect, if this is true, is that we should see that the transferability, so the quality of the train, so the dark green line should start from lower if we transfer from a protein that resembles more what we transfer from. but I don't have any data to support these statements. That's just an hypothesis at the moment.
Evaluate those in terms of the local backbone. I think that you're doing some pooling, right? So I guess you can also have some longer range interaction emerge from the pooling of the local backbone.
So about the which atoms we consider, so we tried with only backbone atoms, backbone and beta, or all heavy atoms, so no hydrogens, but all heavy atoms. So what you're seeing here is results with backbone and beta. And then I'll add some extra information on how we calculate the energy in a second. So we have seen that in principle, the neural network can actually learn even if you go all atom, but it just struggles more and the quality of the models is not as good. So at the moment, what we prefer doing is generating just backbone and beta. And then if we need an all atom structure, we just model in the missing atoms and we energy minimize. About how to measure the energy, if you bear with me for like about 4 more minutes, I'll get to an explanation of how we do it, and that's a quite cool thing, in my opinion. And I lost track. What was the rest of the question?
The question was whether you expect, so when you are only doing local convolutions along the backbone, and you estimate this interpolated energy, whether those local or the energy terms that will change, that you will learn something about, will only be the local structure. So this is related to what Juan asked before, or whether there's also something, I guess, something more emerging from this, and whether these energy terms are, whether there are enough energy terms for them to be physical. I'm not sure.
Right. Yeah. So essentially, what we do, we calculate the energy. So the only position in the neural network where we can actually calculate the energy is at the output. So we cannot calculate any energy in the inner layers because those have representations. We don't know what they are. And we're using a molecular, an MD force field, essentially, molecular dynamics force field. So the molecular dynamics force field essentially describes the interaction between atoms in terms of sum of terms. We have bonds, angles, dihedrals, and those are all connected parameters. But then there's also van der Waals and electrostatics, and those are long-range interactions with atoms that could be, they are adjacent in space, but they could be quite far away in the sequence. Now, since we don't have all the atoms in when we calculate these contributions, we actually do not so we had to prune the long-range interactions because we saw that tended to actually compact the proteins too much, because essentially there's cavities forming if you remove some atoms. So we have the repulsive term of the Lennard-Jones potential, so the Pauli exclusion essentially. And we have not seen much difference in including electrostatics or not. So just for speed, at the moment, we're not including it. One thing I would like to see is if when we actually model all the atoms in, if it actually is a good idea to have the whole MD force field in. But at the moment, it's basically bonds, angles, dihedrals, and van der Waals.
Okay, so I suppose this answers.
No, van der Waals, Pauli.
Yeah, so there's a bunch of questions relating to the local structures, like Ramanujan plots look okay.
Yeah.
And since you're doing convolutions, you can also generalize to proteins of different lengths, right? That's just,
yes.
Yeah. Great.
So I basically the architect, the software that's you can send anything in, and it will just do it. The largest protein we have done at the moment is 480 amino acids. It's actually MRD, but we have not seen much difference. So much difference at which speed it trains. Really, it scales really favorably. And in fact, once the neural network is trained, you can just run it on. And you want just to generate structure, you can run it on your laptop just with the laptop I'm using right now on a CPU. You can generate an interpolation with 20 structures in half a second. We have actually a Jupyter notebook online. If you can play with the neural network. Right. Now, the whole presentation started talking about docking, and we kind of drifted away a bit. But just to bring it back a little bit to docking, the reason why we have the HIV1 capsomer here is because this protein actually assembles into a complex. So we have a crystal structure in the protein data bank of a hexamer of this protein. So we both have a monomer and the hexamer, and the monomer and the hexamer conformation are actually very different. So the question is, can we? And now you see how everything magically ties in. So we have a simulation of our protein. What we can do, we can train the neural network, as I've shown you in the previous slide. And then, as I've shown you in the second slide of this presentation, once you have a representation of the latent space, you can actually use that as a way of generating conformers that the docking algorithm can use. And if you do that, at this point, essentially is, can the docking algorithm pick out from a neural network a conformation that is suitable for docking? And what you're seeing here, that's an overlay between the known crystal structure in gray and the best predicted model from the docking algorithm in red. And there's a couple of interesting observations. So the first one is, now you can see at the top left an overlay between gray and blue structure. If we just compare the conformation of the bound and unbound state of the protein, the difference is massive, 10.4 angstroms, mostly because one domain does a sort of rigid body motion with respect to the other. Now, if we run a simulation and we check whether there is a conformation in the simulation that resembles more the bound state, the answer is yes, there is one. So we are here in a situation, in the where probably the conformational selection is dominant. There's always some induced fits in any docking, but in this case, we have rigid bodies moving. Conformational selection is very present. So we have a root mean square deviation of 3.2 between the best structure we can find in the simulation and the crystal structure of the bound state. Now, the question is, what's the root mean square deviation between the red structure used here by tau and the bound state, the conformation of the bound state? And it's 2.7. So essentially, the neural network could generate a structure more similar to the bound state than anything in the examples, and the docking algorithm could fish it out. That's what we saw in this case. And even that's something that I found even cooler is, in the latent space, where is this red point located? Now, I'll show again a latent space of our P24 on the right side, but now I'm coloring it differently. So in red, projected in this space, those are points that are used by the validation set, just basically where you expect the molecular dynamics simulation to project. And I'm showing just the convex hull of that with this kind of dashed line around. Now, the background color is the root mean square deviation between the protein generated by the neural network and the bound state. So that's a neural network generated on the trained on the unbound state. It so turns out that the bound state is actually located outside of the front of everything the neural network has seen. And so now I need to put a big watch out. So I am not saying that we have a neural network that can extrapolate to discover new states. Personally, I mean, until someone does it, I would think that whoever claims that are probably bending the truth politically correctly. But what I'm saying is that at least we can do a sort of local extrapolation just by generating something that is just outside of the front of what we're seeing. And the model does not break down right away. So we can still generate something slightly outside, and it will still look like a protein. And in this case, we have a situation whereby the bound state is just outside of that front, which is nice to see. So now we're getting towards the end, and I will do something that normally one shouldn't do, which is answer the question, but does it always work? So I asked Lucy, who a brilliant master student in my group, well, I've shown her all this presentation. I was all excited, and I told Lucy, okay, but now, right, it just works. It's amazing. It's magical. And Lucy said, hold my tea. She went in the PDB and found a full protein and said, okay, now let's see if we can predict the switch between two folds. So what you have here is lymphotactin. Lymphotactin 10 and 40, those are different states. And the key point here is that the protein, to switch from one state to the other, needs essentially to unfold and then refold in a different state. It's not just a little conformational change. So what Lucy did is exactly what we did in the previous example. Took all the structures of lymphotactin in the two different folds, run 1 microsecond simulation for each of them, and then train the neural network with all of those pieces of all those snapshots, and then ask the neural network, please switch between lymphotactin 10 and 40 state. So thousands of examples from each of those systems, 1 microsecond each, and it totally does not work at all. So if we look more carefully, what we notice is that there's a loop that magically tunnels through the backbone of the protein to reappear magically on the other side. And if you're interested in knowing why, it's actually quite fun why. It looks like the neural network learns to cheat. So essentially, before I told you that the latent space can be warped. Now, what happens when we are asking the neural network to generate an interpolation between two states? Well, it will generate that conformation, assess the energy, and then use that energy as a way to, in the backprop, right? So try to create a latent space whereby any intermediate has a low energy. The problem is that in this specific case, it just can't figure out how to build a model of low energy. So what the neural network does, it warps the space so much that the probability of us asking it to generate an intermediate between those two states becomes vanishingly small. It learns to avoid being asked the question of finding a good intermediate between the two states. This is why where we literally go in very accurately, very precise, no, we really want that intermediate, this happens. Because the neural network actually learned to avoid being asked this question through the training process. So we know this is happening. We have a couple of ideas how to avoid this. But it is undeniable that here we are asking essentially the neural network to generate something that is way far off anything it has ever seen. Predicting unfolding, my take is that we cannot just do what we've done now. We will need to combine molecular dynamics simulations and the neural network directly in a loop to basically generate dynamically samples from the neural network and bring those samples back for reinforcement learning. And to this point, now we are actually, and this is for those who actually work in the area, well, we are very excited about this. Everything you've seen up to now can be done with a Python package called molearn. It's just on CondaForge, so you can just install it quickly. We figured out how to have a PyTorch talking directly to the backend of OpenMM. OpenMM is a molecular dynamics engine, and that's what we use to calculate the energy of the models we produce. And essentially what we found is that, well, there was a massive overhead in generating the structure from a neural network, writing a file, parsing it into OpenMM, and then calculate the energy. Now Sam has figured out how to pass the coordinates generated by PyTorch directly via the GPU directly to OpenMM. And that has made us gain orders of magnitude. So the very first time when we trained the neural network used to take us 6 to 8 hours, and now it takes us less than 20 minutes. So massive improvement. And it's quite enabling technology for us, because that means that it's really opening up possibility of playing with enhanced sampling. Just generate structures and send them to the neural network and simulate right away. And one thing actually Sam has figured out is how to, since sometimes we are not simulating all the atoms, generating all the atoms with the neural network, it's figured out how to essentially mask some of the terms to basically extract only the terms that are relevant. So we can also play with different force fields now, which is quite fun. And there's a Jupyter notebook that shows you how you can use a neural network. It looks like this. I'm using it mostly for educational purposes because, as I mentioned, we found that projecting in 2 dimensions is a bit aggressive. 4 and 5 dimensions will be better. But if you just want, we just want our students, for instance, to feel how the neural network behaves, you have here basically a widget on IPython. You can click around, and you have an NGL view on the other side that generates your interpolations. You can see there a statistic in 4.46 seconds, 37 structures on my laptop, which is not very good one. So that's where we're at the moment. So hopefully the next time I see you, I can tell you something more about especially path sampling and training with experimental data. Now, thanks to the people who have been working on that. Those are photos of Durham. It's a pretty little university town with a huge cathedral. Come and see it, and see you soon. And thank you for your attention.
Is there anything you can say about the kinetics of the system?
I don't think we can say anything about the kinetics. So this whole approach was born just from the idea of generating conformers for docking. So if we want to extract kinetics, at the moment, the idea is to generate a potential transition path that makes energetically, that makes geometrically sense. and then go with more classical approaches. The difference, of course, is that would be a path predicted by a neural network and not designed by a human.
Yeah, go ahead.
I was thinking that because you impose in both the training set and when you try to interpolate the pinpoints, you impose that basically you draw a line in the latent space and then you put a point in the right. But that's actually imposing a shape over the
Yes.
space. Yes. So have you tried other ways of interpolating pinpoints? Here after a lot of geodesics and it seems like a very natural way to go.
Yes. So that's an excellent point, and the answer is we have not tried yet. And I think to be said is that, you know, the example I've shown with MERD, the Pacman protein, here we have two states. So it seems that there should be a quite intuitive way of arranging the conformation of the protein in space so that you have the two states. Now, the question is, what if you have 3, 4, or 5 states? Is there still a logical way you can arrange them in 2 dimensions? At this point, it gets much trickier. And this is where we find that for MERD, the Pacman protein, 2 dimensions will work like a charm. We have a protein that has 4 states, and there we need to go in 5 dimensions to start getting something sensible.
I also have a question. So this way of interpolating and then evaluating the energy, it's sort of conceptually reminds me a little bit of the notched elastic band method. So have you heard any sort of comments, or have you thought a little bit about the relationship between these two methods?
Funny you mentioned that, actually. So we have thought about that. So there's a colleague of mine who is basically developing an improvement of the notched elastic band. But so the question is, so at the moment, the only thing we tried is, can we identify, so first train the neural network and then identify paths in the latent space using the notched elastic band on the energy. So in this image here, you can see the red trace, that's just A star. So just the shortest path that is biased by the dope score. But I would like to have the notched elastic band implemented instead.
All right, so another question. So when you are doing this biasing with or sort of the sampling, I think it's a very interesting idea. So, but I guess that to some extent you exclude the possibility of taking sort of non-optimal paths, right?
Yeah.
And I think it's a bit related to the question that Juan asked about the geodesics, that you'll have to require some sort of overall topology of your latent space to be fulfilled. So how are you thinking about improving this? And do you think that beyond going like into like different ways of measuring distances in your latent space, you could also think about different ways of encoding your data, right? Right now you're using something which is not really supernatural. I guess I can say this and you would agree with me, right?
Oh, absolutely.
So how do you see, so what do you see as the sort of the priority sort of in moving this forward?
Oh, there's so many. Right, where to start? Well, first of all, how to represent the data? Perhaps I'll start from data representation. At the moment, one of the things that is actually admittedly quite annoying is the fact that we're using Cartesian coordinates already requires us to align the protein first. So we ideally would it would be nice to have an equivariant network instead, which this isn't. One of the reasons where I think it would be particularly important is if you're thinking about going towards training with the conformational space of multiple proteins having different lengths. At this point, I don't see exactly how our neural network will work well. I would imagine that it might easily learn both states, but just chuck them in completely different areas of the latent space without any relationship with them. But if we're going towards learning something about not more the how would you say, not the proteome, because the dynamics of the proteome, or the dynamome, well, the dynamics of the proteome. So at this point, we would need a representation that definitely is equivariant. So that it is for me that would be the priority. About different transition paths. So yes, so at the moment we are, as we said already, that's a very good point, that we are imposing essentially a topology of our latent space by training it by saying that straight lines should be of low energy. So and there was two things to say about that. One is Absolutely true. One of the things we're actually trying to figure out is, can see in this image here, we're using the A* shortest path. We want to see whether actually, despite the fact that we're using straight lines to generate good intermediates in the latent space, what essentially the only thing it does, it kind of simply pushes down the energy of the whole overall structure generated in the latent space, but then perhaps finding curved paths in that resultant space would still give you better interpolation. So that's one thing we're still testing. If that's the case, we can use large elastic band actually, which would give us multiple paths to go from one position to another, going through saddle points. We have done preliminary work on that, but I have no results to show yet. But yeah, so the point on imposing a topology, I think, is really, really, really important. And I don't think I have a definite answer to that, to be honest.
All right. Any final questions? Otherwise, I think we can thank Matteo again.